Everybody, this is the walkthrough video for Monday. It is October 13th. Try one week 10. Still no bell work this week. Still working on grades. Today, we're going to learn about how enzymes work and how they're affected by their local environment. We're going to talk a little bit about why this is so important in a moment. We begin by taking a look at graphs depicting enzymatic reactions. There's a handout for you in the extra pages box for when you return, but we'll also look at those three graphs right now in this video. We then worked through some sample problems about enzyme and cell regulation. If you were absent from class, making sure that you have paid very careful attention to 3.2 and completed the topic questions might be enough for you. If you'd like to review those additional sample questions, they are going to be in the extra pages box after fifth period. Today we finished toothpick ace. If you weren't here, I am going to ask for a completed lab notebook check when I see you next, hopefully on Tuesday or Wednesday. And like I told you at the end of class today, we opened up some AP Classroom topic question quizzes, multiple choice on enzymes and environmental impacts on enzyme function. That's 3.1 and 3.2. All right, in a moment, I'm going to talk a little bit more about this, but to finish the toothpick ace simulation, we're going to identify the independent variable, the dependent variable, and the control variable. That's what you change, what you measure, and what's the same between the two. Our guiding questions, we're still thinking about how enzymes make life on Earth possible. And, a story that we've been telling in APBio so far is that protein activities are really important. You just haven't really thought about it in this way yet, right? Phenotypes or physical characteristics are definitely determined by protein activities, and enzyme activities are a great example of one of those. Are all proteins enzymes? No, some of them are structural proteins, making sure that, for example, cells stay in the same place in an organ. Are all enzymes proteins? No, you've already done the RNA world experiment, and so you know that there are RNAs that are capable of being a catalyst. But for your purposes, when we talk about enzymes, we are talking about proteins. This is the first type of reaction graph that you might see. We saw it in the video intro on Friday. This is sometimes called an energy profile or a reaction profile. And you can see the x-axis is kind of made up. It's like the course of the reaction. And the right-hand scale is usually for the purposes of biology, our biology class, like intro college level bio, usually it doesn't have any scale. There's a starting materials here with the reactants, that's the starting materials, and then the product or substrate, and then the products are down here. Now, what enzymes do is they lower the activation energy. They don't change the amount of energy of the reactants to the products, just the amount of energy that's needed to transition between the two. As we saw in Friday's video intro, oftentimes those substrates end up at an energetically unfavorable steric conflict, like a clash, or like the structure is just very high energy. Enzymes, by protecting that and coordinating the atoms, can lower the activation energy. The same way that the promise of an extra piece of pie might make it so that when you were younger, you and your cousin would have picked up your Legos a little bit faster. The second type of graph we're looking at is a product over time graph. So here are the products. The brackets mean concentration. And then there's time in some sense of unit. In this particular case, the independent variable was more enzyme. And you can see there's a graph for what it was like the first time, and then a graph for the tube that had more enzyme in it, changing that independent variable. And then the third one is changes in a variable. Remember that independent variable is what you test, the dependent variable is what you measure. In this particular case, it says pH or temp. We're looking at a graph where the x-axis each represents different test tubes with different environmental conditions. In this case, it's pH, it could be temperature. And then over here, you're seeing the amount of product after a certain amount of time, or sometimes you'll see it reported as an initial rate of reaction. Both are valid. And you'll see for most enzymes, that this particular one is like the pH of 6.8 is the optimum pH for this enzyme. Why is that pH optimal? Do you remember how amino acids some of them have positive and negative charges? pH can change the positive and negative charges, meaning that they no longer coordinate in the same way, and that changes the shape of the protein. So we worked through this assignment. Again, you're welcome to take a look at it. The extension questions are particularly rich for us, but they are not required. And so at the end of class, we finished the toothpick ace simulation with the independent, dependent, and some control variables listed. No other conclusion is necessary for this, but I do want to see two data tables, one graph with two lines where you've connected the dots and a key, and then the list of variables that's shown up there. Outside of class, if you don't finish them, making sure you are outside of class, sorry, making sure that you've worked through that quiz on enzymes and that quiz on environmental impacts on enzyme function. Not grading those, not checking those, but really helping you make sure that you've gotten that multiple choice practice that's going to be so important for our test. They have just announced when the tests are. It's the same schedule if you've been to South. But your test, remember, will be on Thursday, October 23rd. It'll take all of the class period. And then we'll do our test corrections on Friday. If you're not available, if you haven't taken the test by the time we start the test corrections, you won't be able to do those test corrections. So if something has come up, I know a couple of you are leaving on Thursday, please see me and we'll make sure that we can take care of that for you. All right, that's it for me. I hope you're doing well out there. Really fast week for us. Oh yes, tomorrow we're doing an enzyme lab. You will need to wear goggles. Many people will need to tie their hair back. There's nothing terribly dangerous, but it's really unpleasant to get hydrogen peroxide in your eye. All right, take care out there. See you soon.